2 slices of commercial whole wheat bread release glucose into your bloodstream at a rate faster than 6 teaspoons of pure white table sugar. When that rustic brown slice hits your salivary enzymes, your biology does not perceive a health food or a pastoral wheat field. It perceives an avalanche of fractured amylopectin molecules rushing toward your bloodstream. That golden whole grain stamp on the box promised sustained energy, but your cellular machinery is forced to manage a metabolic emergency. What you are about to understand is the profound difference between the chemistry of an intact plant seed and the biological impact of modern industrial milling. We are going to look past the marketing claims and map out exactly what happens when pulverized whole grain starches hit your digestive tract, how your pancreatic beta cells respond at the micro level, and why the physical structure of your food dictates your cellular health far more than the nutrition label ever tells you. To understand why this happens, we have to look inside a single kernel of wheat, known botanically as the caryopsis. In nature, a grain is an armor-plated delivery vehicle for plant reproduction. It consists of 3 distinct layers: the outer protective shell called the bran, rich in fiber and minerals, the tiny nutrient-dense core called the germ, packed with essential fatty acids and vitamin E, and the starchy reservoir in the middle known as the endosperm. In its wild, unadulterated state, the endosperm's glucose polymers are locked behind a fortress of insoluble lignins and cellular walls. Your digestive enzymes have to work slowly, peeling back layer after microscopic layer over several hours. But that is not what sits in modern grocery aisles. To make modern whole wheat flour, industrial steel roller mills crush that seed with immense pressure and high speed. Even when the millers put every single milligram of the bran and germ back into the bag to earn that legal label of whole grain, the physical architecture of the seed has been completely obliterated. The grain has been reduced to microscopic particles with a massive surface area. Think of it this way: if you light a solid oak log with a match, it burns at a slow, steady temperature for 4 hours. That is an intact kernel. If you take that exact same log, grind it into fine sawdust, and throw a match onto the pile, it does not burn steadily. It explodes in a flash of heat within 3 seconds. That sawdust is modern pulverized whole grain flour. When you chew a slice of whole wheat toast, an enzyme in your saliva called alpha amylase immediately begins cleaving the alpha-1,4 glucosidic bonds of the starch. Because the flour particles are so infinitesimally tiny, this enzymatic breakdown happens almost instantly. By the time that bolus reaches your duodenum, the starch has already dissolved into individual glucose molecules that flood across the intestinal epithelium through sodium-glucose cotransporters. Back in 1981, Dr. David Jenkins and his research team at the University of Toronto demonstrated this exact biological reality when they first developed the glycemic index. They discovered that standard commercial whole wheat bread had a glycemic index score of roughly 71, virtually indistinguishable from white bread made with refined flour, which scored around 73. Both caused a nearly identical rapid surge of glucose into human blood vessels within 30 minutes of consumption. The biological reality is that your enzymes do not judge a food by its color. If you are finding this breakdown of starch surface area and digestive kinetics fascinating, take a quick second right now to subscribe and drop a comment below sharing what whole grain you eat most often. When you engage with these exact scientific mechanisms, it signals to the platform that real human biology matters more than marketing slogans, and it directly supports our work in pulling back the curtain on modern food processing. Now, let us trace that tidal wave of glucose as it travels through the hepatic portal vein and arrives at your liver and pancreas. When glucose enters your bloodstream at that speed, your pancreas cannot afford to be subtle. The endocrine beta cells detect this steep concentration gradient and initiate a rapid, high-volume release of insulin. Insulin functions like biological software, commanding your cells to open their glucose transporter 4 portals, known as GLUT4, and pull that sugar out of the vascular system immediately to prevent toxic glucose concentrations. Your blood vessels are delicate biological highways. Having too much circulating glucose is corrosive to endothelial tissue, so your body will do whatever it takes to clear it. But here is the metabolic trap. When you flood the system with rapid-acting carbohydrates, your pancreas frequently overshoots, pumping out an abundance of insulin to match the sudden peak. 2 hours later, that massive wave of insulin has cleared the glucose from your blood so aggressively that your circulating sugar levels plummet below your baseline fasting levels. Have you ever eaten a large bowl of heart-healthy whole-grain cereal at 7 in the morning, only to find yourself irritable, foggy-headed, and searching for another snack by 10? That is not a lack of willpower. That is reactive hypoglycemia. Your brain, sensing a sudden drop in fuel availability, activates your autonomic nervous system to release cortisol and adrenaline, screaming at you to find fast energy. You are trapped on a biochemical swing set. Soaring glucose, excessive insulin, plunging glucose, stress hormones, followed by intense cravings. Over years of repeating this cycle 3 or 4 times every single day, your muscle and liver cells begin to defend themselves against the chronic, deafening shout of high insulin. They downregulate their receptors. This is the onset of cellular insulin resistance. To make matters more complex, when the liver is overwhelmed by frequent bursts of rapidly absorbed starches that exceed its immediate glycogen storage capacity, it activates a metabolic pathway called de novo lipogenesis. It converts that excess carbohydrate substrate directly into saturated fatty acids, specifically palmitic acid, packing them into very low-density lipoproteins that circulate throughout your vascular system. Let us look at a landmark study conducted by Dr. Karin Hermansen and her colleagues, who investigated how the physical form of cereal foods influenced glycemic and insulin responses in human subjects. They compared intact whole grain kernels against finely ground whole grain flours with identical macronutrient profiles. The biological response was night and day. The intact kernels produced a flat, gentle glucose curve and required vastly less insulin secretion from the pancreas, whereas the milled whole flour provoked an aggressive, sharp insulin surge. The researchers proved that the physical intactness of the plant cell walls, what food scientists call the food matrix, is the true regulator of metabolic health, not the mere presence of ground-up bran. Pause for a second and let that biological truth settle into your thinking. 2 foods can possess the exact same list of ingredients, the exact same grams of carbohydrates, and the exact same fiber count on the back of the box, yet trigger completely opposite hormonal cascades inside your body simply because of how they were physically assembled. This does not mean grains are your enemy. Far from it. Grains possess remarkable micronutrient profiles, unique prebiotic fibers like beta-glucan, and critical phenolic compounds that feed your microbiome and protect your cells. The deception is not the plant itself. It is the industrial illusion that pulverized dust behaves like an intact seed inside human physiology. So how do you harness the genuine biological power of grains without riding the insulin roller coaster? Here are 3 concrete, actionable ways to reprogram your starch intake, starting with your very next meal. First, transition your kitchen from flours to fully intact and cracked grains. Instead of reaching for whole wheat flour products, build your meals around whole rye berries, whole oat groats, farro, hulless barley, and wild rice. When you cook a grain where the outer botanical seed coat remains unbroken, your digestive enzymes must manually work their way inward through intact plant structures. For example, replacing instant or rolled oats with 1 cup of cooked whole oat groats or steel-cut oats dramatically alters the gastric emptying rate. The viscous, soluble beta-glucan fibers form a dense gel in your small intestine that physically slows down glucose absorption, yielding a steady, prolonged energy release over 4 to 5 hours rather than an explosive 20-minute spike. Second, leverage the biochemical magic of starch retrogradation. When you cook a starchy grain like brown rice, quinoa, or whole barley in boiling water, the crystalline starch granules absorb water and swell in a process called gelatinization, making them easy for enzymes to cleave. However, if you take that cooked grain and cool it in your refrigerator for 12 to 24 hours, something extraordinary happens at the molecular level. The amylose chains align themselves into tightly packed crystalline structures that your alpha amylase enzymes can no longer break down. This newly formed compound is called type 3 resistant starch. When you eat this cooled or reheated grain, that resistant starch bypasses your small intestine entirely, causing 0 spike in your blood sugar. Instead, it arrives pristine in your large intestine, where billions of beneficial bacteria ferment it into short-chain fatty acids, primarily butyrate. butyrate directly nourishes the cells lining your colon, seals your gut barrier, and sends biochemical signals back to your brain to increase satiety. Simply by cooking a batch of grains ahead of time, chilling them overnight, and reheating them for dinner, you can lower the glycemic response of that exact meal by up to 20 to 40%. Third, practice metabolic sequencing and macronutrient anchoring. Never introduce a concentrated starch into an empty stomach. When starches arrive in an empty stomach, gastric emptying is rapid, sending the glucose straight into the duodenum for instantaneous absorption. If you instead anchor that meal by consuming a foundation of raw or cooked leafy vegetables, followed by proteins and healthy fats first, you trigger the release of gastric inhibitory polypeptide and glucagon-like peptide 1 from your enteroendocrine cells. These peptide hormones physically slow down stomach motility. If you then consume your intact grains at the end of the meal, coated in a tablespoon of cold-pressed extra virgin olive oil or paired with an organic acid like raw apple cider vinegar, the acetic acid slows the activity of salivary amylase and further delays gastric emptying. You effectively transform what could have been a sharp blood sugar peak into a gentle, rolling metabolic wave. You will never look at a loaf of bread, a box of crackers, or a breakfast cereal the same way again. You no longer view food as a collection of passive calories or superficial marketing claims printed on cardboard. You now see every meal as biochemical instruction code, a precise structural input that directs your enzymes, commands your hormones, and programs the metabolic fate of your cells. You are in complete control of that biological software. What whole grain is sitting in your kitchen right now? Hit the like button on this video right now as a practical bookmark so you can easily reference these exact cooking and cooling protocols next time you are preparing your meals. We are already deep into the laboratory research for our next investigation, breaking down how a common morning beverage radically alters your cellular hydration and cortisol rhythms within 90 seconds of your first sip. Subscribe to Nutrition Decoded right now so you do not miss that discovery the moment it goes live. Your biology deserves the truth.